The central dogma says that the instructions for making an organism, the instructions for putting together a cell and what makes different tissue types different from each other and how you construct those tissue types to create organs and how you arrange those organs to create an organism, that is stored inside DNA, deoxyribonucleic acid. And when you use DNA to make more DNA, that's DNA replication. And remember all those enzymes we talked about, right? You got DNA polymerase 3, DNA polymerase 1, you got ligase, no particular order, by the way, helicase, topoisomerase, primase, and who am I missing? Oh, single-stranded binding proteins. Right, all of those different parts, you got the leading strand and the lagging strand, that all takes place in the nucleus because DNA stays inside the nucleus. So DNA holds the genetic instructions, and we've already done the genetics section with you guys, so you guys know all about how different alleles work and how different versions of a gene can code for different things. But if you actually want to be able to use that information, the information can't be stored inside the nucleus. It's actually, you got to take it out into the rest of the cell and you got to use it to make a protein because proteins do all of the work inside of your cell. It's one of the first things I taught you, I'm pretty sure. Like in the first couple of weeks of biology class, we said proteins do everything interesting inside of a cell. So how are we going to get it to protein? And I'll make protein a purple protein, keep the P's, right? Good brand integrity here. So we have protein. That's the actual functional part. So the DNA is the information holding part, but the proteins do the actual work. And the DNA just holds the instructions for making proteins. Now, what is a protein? It's a polypeptide, right? A polypeptide that has been folded. This computer is a little bit laggy, but for some reason it seems to have a bias for not registering horizontal lines, because I'm noticing that a lot of times the tops of my letters are getting left off. Like right there. And what is a polypeptide? It's a chain of amino acids. Held together by peptide bonds. Those peptide bonds are accomplished by dehydration reactions. Remember way back when we were talking about about monomers and polymers, we string together these monomers, the amino acids, to make the polymer, the polypeptide, and then that polypeptide will spontaneously fold to get a protein, and that's actually functional. But these two molecules don't speak the same language. DNA is made out of nucleotides, proteins are made out of amino acids, so we need an intermediary. We need a translator. And that translator comes in the form of RNA. Right? RNA is a nucleic acid like DNA, but it's a lot more flexible in its functions. There's lots of different kinds of RNA that we've talked about. We've talked about mRNA, and we've talked about tRNAs, and we've talked about rRNA, the stuff that you find inside of the ribosome. We talked about snRNA when you are taking the, well, I guess we haven't talked about that part yet. That's after transcription. You have to process the RNA, and some of the little bits get snipped out, and that's accomplished by small nuclear RNAs, snRNAs, as part of the spliceosome, which is a fun word to say. So information in DNA gets copied into RNA, and we call that transcription. Okay, transcription. And if you guys have already started watching some of the videos, you'll know some of the things that are required for transcription. You have transcription. Let's abbreviate factors. It's getting really laggy on me. Transcription factors. You have another enzyme called RNA polymerase. Technically, it's RNA polymerase 2, but we are only talking about one of them, so we'll just call it RNA polymerase. Whereas in DNA replication, we had two different versions of DNA polymerase. So transcription is taking DNA information and using it to copy into an RNA molecule. So we're making RNA. What enzyme do we need? RNA polymerase. DNA replication, we're copying the DNA. So we're making more DNA. What enzyme do we need? DNA polymerase. So remembering this central dogma chart can be kind of helpful to remembering which enzymes go with which step. And you also have stuff like the promoter region, which is the beginning part of any gene on the chromosome. And you have the terminator region, the terminator DNA. Okay. All of that is part of the transcription material. But then we take that copy of the information and we have to translate it into polypeptide language, and we call that translation. DNA is your hard copy of the gene. It's the original, right? Sometimes you have certain documents that you want to keep the original, like for example your social security card or something like that. You want to keep the original nice and safe. That's locked away. But sometimes you have to, when you're applying for a job, you bring it into human resources and they have to get like a little photocopy of your license or your thing. That photocopy is our RNA. In this analogy, the original is the DNA, and you want to make sure that the DNA stays safe, so it stays bundled up inside the nucleus, not allowed to leave. It only gets to leave once during mitosis, but at that point it's wrapped up so tight in those little chromosome packages that it actually can't be used for transcription. So we need to copy the information in the form of RNA, and if something happens to the RNA copy, no big deal. We'll just transcribe another one. So that can get damaged, and if it does it wrong, it's fine. We have the original still that we can check. If anything happens to the DNA, well, you can get mutations, and then every single time we transcribe that gene afterward, it's going to be the wrong sequence of bases. So you want to keep the DNA safe. It's kind of like a central archive with blueprints for making polypeptides. And then the RNA is just a copy for one time. We're going to make this polypeptide. We're just going to take it, send it out to the ribosomes, and tell them to do their job. And the translation happens at the ribosomes. Ribosomes. Look at that word again. Maybe it was a different class. Some, whenever you see some, that means body in Latin. So when we're talking about body cells as opposed to like gametes or sex cells, we call them somatic cells. They're just part of your body that's like skin cells or bone cells or muscle cells or anything like that. A chromosome is a body within the cell, an object within the cell that takes up dyes very well. So it's the color body, the chromosome, right? Lysosomes are little objects, bodies within the cell that break down materials. They do lysis. So the ribosomes are bodies within the cell that contain a lot of RNA inside of them. The form of RNA that you have inside of ribosomes is called rRNA. This RNA here, one sec. This RNA here is called mRNA, messenger RNA. That's what we're creating during transcription. But then in translation, we need to make a polypeptide, and that requires a couple of different forms of RNA. One, we need the mRNA, the ribbon we just made, and we need the rRNA in the ribosomes. The rRNA is the part that actually does work in the ribosomes. It's what's actually holding things together and catalyzing the bonds between amino acids. We also, get to my purple color here, we're also going to need tRNAs. tRNAs bring new amino acids to the ribosome one at a time, and they also have something called an anticodon on them. Which reads the three-letter codes on mRNA, which is called a codon, right? So mRNA has codons, whereas tRNAs have anticodons. And they're just called codons and anticodons because they match up with each other. They're compatible with each other. Okay? And a codon is just a sequence of 3 nucleotide bases. So like A, U, G, that would be a codon. The tRNAs would read that, and then they would know which amino acid to bring to the ribosome in order to make this polypeptide chain here. So you need ribosomes, you need the mRNA, you need the tRNAs, you're going to need the amino acids, of which there are 20. And this is also the area, this is also the section where we have what are called stop codons. Okay, so that's where translation will stop when we reach a stop codon on the mRNA molecule here. So that's the central dogma of molecular biology. DNA is transcribed into RNA to mRNA, and then that mRNA is translated into polypeptides, which then fold to become proteins. There's one additional step here, which, nice, how about a nice saturated green color. There's a step kind of in between here, which is called RNA processing. And the reason why we need that step is because some of the information in genes is not necessary to making the polypeptide. Some of it is not what we call non-coding DNA. And when transcription goes through, we copy the entire gene, even the stuff that we don't need. So we have to cut out the extra bits that are not required. And that's what RNA processing is for. So in RNA processing, we remove the sections of the mRNA molecule that we just made that do not code for polypeptides. We call those introns. So introns will be removed. And then we splice together the stuff that remains, which is the exons. Okay. These are just names for different sections of the RNA molecule. So exons spliced together. And then, in order to make sure that we don't lose a few bases off the end of the mRNA molecule, we add a kind of container for it. We add a little safety equipment, which is called the 5' cap and the 3' poly A tail. Poly A because it's just a series of adenine nucleotides. A, on and on. Could be hundreds of A's long. So it's a nice long tail, and that just helps give a little bit of directionality to the mRNA molecule. We know which end is which, and it also makes sure that it's safe during transport here. So that's the central dogma of molecular biology and how DNA, RNA, and protein kind of relate to each other.